Okay, so we're going to talk about how stuff transports across that cell membrane, so into or out of a cell, right? There are a couple major categories we're gonna break into and think about. So here's our intro. So number 10 in our notes, that's where we are. It says, what is diffusion? So this is just when something, a particle in a solution, right, moves from high to low concentration. That's all it is. It's something going from here to here. It's just spreading out, right? It's when until you reach a point of equilibrium. So it's like when you put a drop of food coloring in, I don't know, a big glass of water. That food coloring is really dense, right? High concentration of color, and it slowly spreads out until you have a kind of even mixture. All right, so I always talk about diffusion in the context of fart particles, like farticles. So if someone on one side of the room rips a big nasty stinky fart, one of those real greasy ones, they get stuck on in like your hair and on your clothes. You can like taste it. You know what I'm talking about. Well, the first person to smell that is the one who dealt that. Oops, sorry. Anyways, whoever smelt it dealt it. But the next person to sniff it is going to be the nearest person. Eventually, everyone in the classroom is going to smell that nasty, stinky fart. And that's when the farticles, will have reached the point of equilibrium. All right, so on number 11, we're talking about something called osmosis. All right, so osmosis is really a type of diffusion. It's strictly about water, though. So it's diffusion of water across a membrane. So we say selectively permeable. That just means something that is selectively permeable lets some stuff through and not others, right? So the screen in your window is selectively permeable, right? It's permeable to air. It's not permeable to owls or bats or orangutans. Lots of things can't fit through there because they're too big. But it's permeable to air. Okay, that's what that means. Okay, so we're going to talk about this. You could get plenty of practice about this, but we got to move on to 13, 14, 15. They're all kind of together here. We'll talk about relative concentrations, right? So if you imagine a cell and an environment around it, you have to think about the concentration of stuff in the cell versus the concentration of the environment it's in, all right? So we're going to be modeling this with our egg lab, and more on that soon. I'll introduce you to that soon. All right, so hypertonic. Hyper means more than, okay? Tonic refers to the concentration of stuff in the water, so how much solute there is. It literally means above strength. Hypotonic means below strength, so less than. Isotonic means same strength. So we're talking about concentration of the solute inside our cell versus outside. That's what these terms apply to, and I'll explain how those work coming up soon. All right, so this stuff might be a little bit hard to wrap our brains around. That's why we'll get some practice coming up. But I want to explain and show you what hypertonic, isotonic, and hypotonic look like. So if you have a red blood cell and you put it in a hypertonic solution, that means there's more solute out than in. All right, that means there's more water in than out. There's more water inside the cell, and the water escapes and leaves that cell, leaving it all kind of shriveled. All right, so the net movement of water in a hypertonic solution is out of the cell. Isotonic, same, equal, both directions. Hypotonic, here on the right, that means there's less dissolved stuff in than out. Okay? No, that means there's less dissolved stuff out than in. I even said it backwards, which means the net movement of water is going to be from high to low concentration. More water out, less water in. Water comes into the cell. Now, that's a lot and it's pretty fast, and I know, and we're going to get some practice. So look at the images. They do a good job of kind of illustrating what's going to happen in a different medium or solution. All right, and so we'll get some practice with this and we'll make sense of it. All right, so let's talk about something called 16 called facilitated diffusion. So again, number 16. So this is where we have those protein channels we diagrammed before and labeled. Sometimes you have big particles or charged particles or something that wouldn't otherwise be able to diffuse straight across the cell membrane. So you just basically need to have a door open. So facilitated diffusion is just a doorway, right? That's all it is. So big particles can move through these protein channels. So if we facilitate, we help the process of diffusion by just creating a channel for stuff to flow through. Again, if it's diffusion, we're talking about high to low concentration. So lots of stuff here will on average flow that way if there's less of it here. There it is. Okay. All right, so far we've talked about processes that happen all on their own. Diffusion, right? Simple diffusion. We've talked about facilitated diffusion. We're not moving particles around, they're doing it on their own. But we also have something called active transport. So this is usually against the difference in concentration. So usually stuff goes from high to low concentration, that's diffusion. Well, what if you want to concentrate stuff? Take it from low concentration, squeeze it all together into high concentration. We call it active transport. And the key with this is it requires the active use of energy. So all that ATP we cranked out there using our mitochondria back in cellular respiration, we're going to be using a bunch of that now to help with things like active transport. It takes a lot of energy, right? So here's the analogy I like to use. So let's imagine you got chickens. You got a bunch of chickens. You got a nice big old yard and you got chickens. Well, at night, you got to get your chickens back in their coop, in their little cage, so that they're protected from dangerous owls and raccoons, right? Save them in a fox or a coyote, who knows? So it takes some energy to get them back concentrated in because the chickens were running all around the yard. They love it out there. They're loving it. And you got to go chase them down. It takes energy for you to go chase down chickens, put them in the coop, run after chicken, grab chicken, put chicken in coop. It takes energy for you to take them from low concentration as they're all spread out in your yard to getting a real concentrated and focused in that coop. It doesn't take energy for them to spread out. So in the morning when you open up that coop, they just kind of work their way out into the yard and spread out. All right, so this is my perfect analogy for you. You're welcome. Let me show you, here it is, what active transport looks like. So we want to go from low to high concentration. You can see on the top of our diagram, we only have 3 molecules, and on the bottom, we already have 5. So we have fewer on the top than we do on the bottom. But we want to go towards where more already are. Well, it takes energy. So we're going to apply energy, boom. It's going to change the shape of our protein, help us pull a particle through. So that's active transport. Go from low to high concentration. Okay, and then the last kind of topic to discuss about coming in and out of a cell are these larger amounts of items called endocytosis and exocytosis. You already know endo means inside and exo means outside. So if you're bringing large amounts of material into your cell, it's called endocytosis. Side note, if you're bringing in liquid, it's different than if you're bringing in solids like food, basically. So this is on a, again, cellular scale. We have phagocytosis and pinocytosis. That's a why, just for you. You can go impress your friends now. Exocytosis is when you take these large amounts of material and remove them from a cell. Okay? That's that process.